Any question? Oh, all right. Okay, perfect. Thank you. And to give you an overall introduction into the graphic user interface, and then one major point is how to load data into InstantClue, which are maybe the objectives in actually getting started. Then what kind of different chart types we have in InstantClue, how to modify them. I will probably in the seminar mix up these points a bit, and but overall we will cover these. Then there is a so-called feature, which is the main figure, which allows you to arrange your charts actually and export them, which minimizes the work that you have to do afterwards for alignment and resizing. And then we will cover various options to filter your data based on categories or on string searches or just numerical data. And then maybe, I don't know if we have to, but there might be the opportunity to make a small break, just 5 minutes or so. And then we will dive into a bit more advanced plotting techniques, which will cover hierarchical clustering, k-means clustering, dimensional reduction. I will probably not go into the detail into these techniques, how they work, and the mathematics and the theory behind that, because this is really more about the software and not about the statistics, statistical background of these. But if you have burning questions, just ask me. Yeah? Okay, then just to basically introduce a bit these data formats that we are dealing with that are implemented in the software, which are available as a tutorial data file, which will always, or most of them will look like this, where you have some kind of gene or protein in rows, and then you have certain quantitative values, which are coming from the mass spectrometry-based proteomics, so meaning the abundance of the protein in your different samples. And then we most of the times have something like a log2 fold change between two conditions that might be Y type, control, whatsoever. I will go into detail once we are talking about this tutorial data. And then you have a minus log 10 p-value. And this is usually coming from a t-test. A t-test just simply compares two groups and checks if the mean is different between these two groups, and then provides a p-value, how likely that is to happen by chance. And this we can use as a readout for significance. And as you probably know, the p-value usually varies between 0 and 1. And the most interesting proteins which have a very high significance would have a low p-value. However, if you plot it, then you will basically have the range between 0 and 1. And the most interesting part, meaning the proteins or the features that are having a very low p-value, will be squeezed in a very small part of the graph. But if you're using the minus log 10 transformation, then basically these very low values become very high and are just visually more appealing. You will actually see what I mean by this in a second. And then what we usually have, at least in our workflow, is a column which indicates by a plus sign if this is significant. Probably some of you have learned that the p-value has to be between below 0.05. However, if we are dealing with very high dimensional data, meaning we have thousands of proteins, and we repeat this test, this t-test, then we run into a problem which is called the multiple testing problem, because we simply repeat this test and then 5% will be basically significant even if we invent random data. And to basically circumvent that, a lot of people using the FDR, which is false discovery, and you can control that to a certain percent, and very commonly we control it to 5%. That's why it's written here. If you want to know more about this FDR, please contact me and I can explain it to you in a bit more detail. And before I open it, finally, the software, just one more slide, and that is dealing with so-called column data types. And this is just general for data, how they are structured in the software. what you will see is that in the end, we are just working with the column headers. So we don't have a spreadsheet just per se like in Excel, but we're working on the column headers, and these column headers are divided into the different types. So for example, you can have string data, so meaning just text data like a G name or some annotation, some group, knockout, wildtype, and so on. Something written in text, which is all called categories in InstantClue. Then we have integers, which could be a count 1, 2, 3, 4, 5, anything that doesn't have a decimal point, and then everything else, which is then a float, is having a decimal point, as you can see here, these examples. And these are the different types and functions that are available in InstantClue, sometimes depending on the type of data. So, and as has been pointed out already by Daniel, I guess, is that there is a problem in the current version that you can download from GitHub with the dark mode. So if you want to use it, please use or switch it to light mode for the moment. But I hope we can fix this in the very near future. And yeah, but for the moment, please do like this. Also, if you have an M2 processor from Mac, you might run into problems, but this will also be fixed very soon. And just for your information, so this is how the GitHub page looks like. If you Google for InstantClue and to download the latest version, you can go here to releases, and then you can see actually the latest version. You can see already that here's a draft for the version that we are using. So we will upload it relatively quickly. If you have any issues, you can just go here to issues and then open a new issue. Best would be if you simply describe what you have done and on which operating system you are, and then that's it. And also, sadly, not as extensive as I wish it would be, is here the wiki, and you can find here this tutorial here, and I will post then also the link of this recording wherever it is hosted, and probably we can also link this page to the video. And then I will quickly start with the general introduction to the graphic user interface. And so the main function, so I have to rearrange a bit your videos. Okay, so this is how it should look once it has started. And let me just quickly go through the main windows. So on the left hand, we have the so-called data preview, and we will see once we uploaded data, we will see all the column headers here divided into the so-called numeric floats, integers, categories. And in this window, we can actually upload data, we can load a session and download and save a session. We can have a kind of a spreadsheet-like view when you click on this, but this I will show you in a second. We have here another window. So the first one is the so-called quick select, which we will go through extensively, which allows you to perform kind of visual analytics to find certain rows in your data, to mask data and so on. So the functionality is relatively extensive, and we will spend some quiet time on this because it's has been proven to be relatively useful. Then we have the so-called live graph, which can, once we hover over a certain graph, can show you additional data here. Then we have an analysis tab where we can perform statistical tests between two conditions in the graph. And this we will get to know in a second. Most importantly, we have, close this one, we have these, which we call so-called receiver boxes, which are there. It's one for numeric data. So anything that has a float or an integer can be dragged and dropped from this tree view to this one. And this will basically trigger a plot. And just to give you a very short introduction in how this works, is you can always go to File, Load Example, and then there's Tutorial Data 2. And as you can see, this loads just the column headers, which you can see here. You can go on this one, and then you can see like a spreadsheet. So what we have here is several conditions. control 1, 2, 3. These are the samples. We'll just quickly go through this. We have certain quantitative values. We have missing values. And then we have some kind of gene names. So in each row, there's a protein group. And in columns are the samples, as I told you before. And just to get started, actually, you can just drag and drop your column header from here to this numeric, and it will plot scatter plot. You can always change the chart type here on this right panel. So for example, if you want to have a box plot, you can just click on box plot or bar graph, and you can switch between them relatively easy. If we want to add a new one, we can just add this one, and we can add more. So you can just drag and drop, or the other possibility is to just go to this plus sign here, and then you can plot these distributions. A box plot simply shows the distribution of the data. So it aims to summarize how these values that you have in this column are distributed by describing the data using a box. And this is usually the 75% quantile. This is the 25% quantile. This one is the median. And these are, the median is the value where 50% of the data are below this value and 50% of the data are above this value. And then you have minimum and maximum, and everything that is here considered across is considered an outlier. And this is defined usually by 1.8 times away this box height from the median. You can set all this, and you can also read up again what are these, all these lines are, these whisker plots, but they are very useful in showing you the distribution of data. And it's usually a good idea to compare all the, if you have proteomics data or transcriptomics data, to compare the distribution across all these samples if they are more or less similar. Yeah. Okay.
Hendrik.
So the outliers are set using the top part there where the mouse is?
Yeah, so everything this is indicated by cross is considered an outlier in this data, and they are defined usually by taking the interquantile range, which is from the 25% quantile to the 75% quantile, and then if the value is 1.8 times away from the median times this interquantile range, then a box plot considers this as an outlier. Yeah.
Yes, and how about I change the 1.8?
Yeah, that is possible actually. So you can just go to this settings. You can go to box plot, and then so you can basically set here, I don't know, 2.0, and then basically it should consider only outliers that are away 2.0 times the interquantile range.
All right.
Okay. And Tanya, I think
at this point, if you see that there's one sample that is very different from all the others, then you exclude from your analysis for the.
Yeah.
How do you proceed? How do you?
I think this is a bit
opinion?
Depending on what data we are talking about. At least in proteomics data, this would indicate that there is a significant less number of proteins identified, because the software, how it works, I mean, this is a bit specific now, it will try to correct for this minimum number of proteins by shifting them up, since the algorithm always believes that most of the proteins will remain unchanged, and then usually it overestimates the intensities a bit, and therefore you see these outliers. So often it's the reason behind that is that there are less proteins quantified in this, and if this is too extensive, then you should probably exclude it or try to normalize this by other means, because then this algorithm, which is called label-free quantification, I think this is a bit too much detail now, is not suitable anymore, and it will not be able to deal this data if the number of proteins is significantly lower in one of these samples. Yeah.
Mm-hmm.
Yeah, but
okay, but in this example that you give here, everything seems equal, so you would consider everything.
Yeah, exactly. I mean, this is an IP even, so it's a bit different, but yeah. But of course, you should always plot all of your samples. I mean, this is something like this, and you can indeed see that there is some variation here. Yeah, I mean, that is usually this data set is, I think, it's relatively old. So the machines and the workflows and so on have become much more reproducible, and usually we don't see this much of a change anymore in proteomics data. Okay, so but I was just introducing a bit more the software, so just very general, not to go too much into detail into the data. You can remove any column header that you have put here into the box and into the receiver box, I'm sorry, by just right-clicking on it, and it will just replot the same chart, just excluding the data. You have here the colors, and you can also identify the box plot by just left-clicking on the colors. It is true for almost everything that you can change the color here. Speaking of colors, you can also adapt the color palette that you want to use here by just clicking on color, and then you can go here and use blues and so on. So you can just change it, and you will change it for all the upcoming graphs. Yeah. Okay. If you want to rename a column header, you can just double click on it and give it a smiley or something like this. Yeah, so you can just I just double click on it and then have it. You can also remove it by clicking on this X. Be reminded that there is no going back, so you would have to reload the data. Okay, so this is what we can very easily do if we can upload the data, and that is sometimes the detail in Devil is getting the data into, especially if you're from Germany, you probably have decimal points divided by a comma, and this is already a problem actually for the software. And I want to show you some examples how to get your data, because now we loaded a simple tutorial data, which will always work. I have just created here random data in this Excel sheet, and we can really, the easiest way to get data into is to just put Command C and then Command V, and there you have it. Yeah, then you have them reloaded into the software. You can check them here. A good check is always to see if the type is correct. So let's have another example. For example, here I made the decimal point a comma, and if we load now this into the software, it actually thinks this is a text. Yeah, because it just doesn't know what to do with this comma there. And of course, we cannot do any plotting. We can even try maybe to change this data type, but it will say it failed. Yeah. So this is not, this can be very frustrating, I would say, and it's sadly right in the start when you want to use the software. So I want to show you how to change that. So if you always have this decimal points divided by a comma, you can go here. Sorry, I will go a bit slower. You can go here to the right bottom corner, and then you have the settings. And here you have the settings load data. So you can navigate in this drop-down menu to load data. And here you can set basically any parameter for loading the data. So for example, we can here load file and then decimal, and we can change it to a comma. And we don't have to press save or so. We just close the window, and it should be still in my clipboard. And you can see now, if I paste the data in, now it understands that this is really a comma, and that the data are comma, decimal points are comma divided. So then InstantClue basically understands it and puts this in the correct column format or type, sorry, and then we can also use it for plotting. Yeah. So that is what we can change here in this. Another thing is that if you're working with Excel and you are computing something, then there might be something like an error, which is called Wert, if it's in German, or any other type of string that Excel puts out as a warning. or divided by 0. I don't know what is the arrow in German for that, but there are different kinds. And if we now put this one into the software, it will also not, I mean, think that this is actually a text format because of this arrow, which is here called BERT. And conveniently, we can define a set of strings that the software should consider as an NaN, not a number. We want simply to ignore this string. And what we can do is we can go here to the setting, and you can see here where there's an option which is called load file NA values. We can just put as many as we want, and we can divide them by a semicolon. So for example, we could write there, no? We just simply close it. And if we paste the data in, well, doesn't work. Let's see if I did a mistake. Okay. That's a bit of a sad thing. What do I say? I will double check and then write on the GitHub page why this is not working.
I think this is because you changed the decimal point thingy.
Ah, very good. Thank you very much. If you load the data in the correct way, thanks, Daniel, then it works. And this BERT is then considered as an NaN. Yeah. Thank you very much for this comment. And
I also have a question for that.
Yeah.
Can I actually just use a regular expression, because it seems that Excel always uses hashtag some word exclamation mark?
Sadly not in the official version. In our development version, yes, and I will hopefully change that you can write any regular expression and then consider this. Yeah.
Okay, thanks.
Yeah, a good point. I will write it down and then change it and add it in the GitHub page once it's available. Okay. Okay, cool. And this is how you can do that. Most of the things, if you have some characters that cannot be interpreted by the UTF-8 encoding, and the software says, yeah, it cannot read this character, then you can try a different encoding. Probably the best one to try is this ASCII. But this is a very special case. And please, if you have a problem like that, post it on the GitHub or write me an email. Send at the best this file actually as well, and then we can try to figure out how to define the settings to get you going. Okay. Good. I think.
Sorry, I'm lost with what is GitHub page?
This is what I showed to you here. Basically, this is where the code of the software is hosted. You can, if you once created an account, you can create an issue here. So you just click on issue and you would say cannot load data, and then you could describe what data you have and what settings you tried, and then I get an email and I will respond to that and then can help you. So don't just quit the software and yeah, and then stop doing it, but just let me know. Yeah. Okay. Also, in general, if there's a crash, which will probably happen, then just please write me, because then we can actually, or we cannot find all the bugs, and it's very good if you help us. Okay. Now we want to have a little bit of interactive charts now after all this talking, and I will just load the tutorial data 2 again to show you a bit the functionalities of different chart types. So the easiest way would be to start with scatter plot, which just plots two numeric column headers against each other. A special type that we are using is the volcano plot. The volcano plot is using the log2 fold change on the x-axis and plots the data where there's a -log10 p-value of a t-test. And as I explained to you before, this is very often used actually to visualize proteomics data if you have two groups. And I'm just using this as an example. So we are plotting the log2 fold change of this against the -log10 p-value, and by default we get this box plot. we can just click on the scatter plot, and then we get this this view. This, what I will explain now, works for any scatter plot. Yeah, it's not limited to volcano plots. Yeah, and also not limited to proteomics or omics data. You can just plot any kind of data. So first of all, you can see that if we hover over these data, we can see that there somehow it recognizes these data, but and colors them in red. But this is not giving us any information. So we have to tell the software what we want to see. And at the best, it's some kind of text. So let's say we want to see the genome, which is associated with the underlying points. So what we can do is we can just take the genome and drop it to the tooltip button here. And now if we go over the data, then we can see actually the genome. Yeah, so it doesn't automatically recognize genome at a very useful column. We have to tell the software which one to use. We can, of course, also use protein names. We can also use multiple names together. Yeah, and then if we go over this, we can see the information behind them. Of course, this is nice, but sometimes we want to label them, and we have to again tell the software what we want to use for labeling. So if we now go over this and we click on it, then it labels the genome, because as you can see here, I selected the genome and used drag and drop to assign it to the label function. And then we click on it, we can just simply label the points. We can move them around. Be aware to always pick the left bottom corner, which is more, yeah, it works usually the best. And then we can just assign these data points to the name. Yeah, this is already okay. We can label now the data, and we can also, if we go right click here, then we have also access limit. In this case, since it's a log2 project, it might be useful to center the data around 0. So there are some shortcuts that we can use. Then we may want to add some color. So let's say we have, in this case, as I mentioned before in the presentation of the data sets, we have here a column which is called t-test significance, which indicates by a plus sign if that protein is significantly changed at a certain FDR. So here we have plus signs and minus, and we could use that actually to visualize and encode them by color, which are the most interesting ones, because the significant ones for us in this data set are the interesting ones. So what we can do is we can just take it and bring it to color, and then the software labels now the data by the categories, meaning by the data that are in this column. In this case, we have minus and plus, and therefore it labels the data depending on this. Of course, if you have now an additional one, let's say, I don't know, plus plus, then it would open up a new category and label them, of course, as well. Yeah, so it always depends on the data you have in there. We can also make combinations of different columns. Let's say we want to label the combination of these two columns, whatever they are. So we take them, we both, so I just put on Mac, I just put command, and then I have selected them both, and then I put them to the color button. Then I can see that the software finds all the unique combinations, meaning since we have only minus and plus in these columns, we have minus, minus. In these brackets, it indicates the number of data points that are associated with this distinct combination. Then we have minus plus, which is then corresponding to minus in this and plus in this column, and so on. Yeah? So you basically have all these combinations. You can again basically color them in different colors. So either you make them gray by clicking on it, or right click will then open up this window, and you can select the column that you want to use. Yeah? You can always add the legend to the graph. So for example, I just right clicked here and said add legend, and then I can decide where to put it. For example, here upper left, and then you can export this figure to a PDF by just clicking here on this floppy disk, you can select what you want to use. And in this case, maybe you want to use PDF, and then you just save it, and that's it. Yeah, and then you have it, and you can edit it also in Adobe Illustrator or whatever you're wanting to use. Any software that can handle vector graphics is able to handle this kind of data. Okay. Of course, we are not limited to the color encoding, but we can also encode the size. So, for example, we could use now the p-value to encode the numeric value by its size of the scatter point, and for that we simply use again the column header and drag and drop it to size, and then you can see that the size of the scatter plot is now corresponding to the value that is associated here with this -log10 p-value. You can see for numeric data we get a different legend, and this covers usually the same matrices that we also have in the box plot, which we explored already, which is the quantile, the median, and so on, the maximum. And it's interesting to note that if we do that for color as well, then we cannot change the color, yeah, because then it's a fixed one, and we would have to change upfront the color map, yeah, and then it maps it to all. So there we are a bit limited in the range of what covers the color. Okay.
If I may quickly interrupt, there are 2 questions in the chat.
Yeah, I will show. Yeah, I think this is answered, or did you miss it? I think you probably did. Okay.
You said you can export by PDF, but this PDF could be edited directly in Illustrator, for example. You don't have like PNG, APS, like this kind of extensions.
Yeah. If you use PDF, then you can open it in Illustrator and edit it as you want to. Yep. Yeah. Okay, so how to add the legend is you just simply right-click on it, and then you choose where you want to put it. And in this case, I choose center left. Thank you, Vivian. Yeah, yeah, and this is the same also for the sizes. Yeah, you can just put it like this. And okay, if there are no further questions, this is more or less what you can do with scatter plot. I think very quickly you can generate an overview of your data. You can, of course, do the same for multiple scatter plots. So we can plot here, whatever, these ones. Yeah, and then whatever.
So another question.
Yeah.
In the chat.
Okay, yeah, I will show you. I will first finish this and then answer this. So if you have multiple ones, then they are all connected. So if you hover over these data, then you can see where this point is actually in the other scatter plot. So if you have multiple volcano plots, for example, you can you can directly see where the other protein is. However, a problem is that if we now put this one here, then this is true for all scatter plots. Yeah, so simply because otherwise we would have to have a color encoding for each individual scatter plot. And before I go on, I quickly check if I use the right for the volcano plot. I think since some people here are really from proteomics background, I will show this in more detail. So now we have 2 volcano plots, and we would like to assign the significance that are corresponding to these volcano plots, because for this pairwise comparison, as coming from a t-test, we have different significant levels, and there is a way to do this, which is called enable volcano plot style. I just simply right-click here and say volcano plot style, and what it wants now from us is that we have 2 plots, and it wants 2 significant columns from us. So we can just select the corresponding one, these identified by the name here. And if we now do this, then the color encoding is simply saying significance up, down. And this is a special type for this volcano plot where we can then very easily create multiple volcano plots next to each other. If you also label here, then they are labeled in both volcano plots. You can turn that off by saying disable annotations in all subplots. Then the labeling is only done in the plot where you actually clicked. And before I forget that, I want to show you from the Excel file. So if I understand you right, you get an Excel file from the proteomics, and now you want to load that into InstantClue. So you can, of course, simply use the copy and paste function that I showed you to load the data. But of course, you can also go here to this plus sign and then select an Excel file, which then gives you all the sheets that are in this Excel file, and then you can select basically the one that you want to import. So let's, for example, load simply the first one of this Excel file that I showed you, and you can see that then it loads simply this sheet. So if you get an Excel file from your proteomics facility, you can just upload them. Be aware that I'm not 100% sure. I think you can define the setting how many rows are skipped. For example, if the proteomics facility adds a certain header that gives you some kind of explanation what is in this column, then you should ignore that. Otherwise, you get that as a header in InstantClue. But otherwise, there's no limitation for that. Yeah, okay, then cool that there are so many questions. So just to answer the last one, that was if you have two plots, how you can basically align the axis. That is quite easy because you just click on the right click, then opens the context menu, and then you have different options. So for example, you can say set equal X and Y axis in all subplots, and then you basically align then the x-axis in these different plots. This works for all the different chart types that we have here. Yeah. Yeah. Okay. Okay, good. And there are no further burning questions to the scatter plot. Then we may go into the main figure for a short time, because we talked already about exporting the data. And a problem is that if you do the charts now on your computer, on your laptop, and you export them, and then you come back to the lab and connect the monitor or so, and you export them and maybe as a PNG, then the resolution will be different because it depends on the screen size. So what you can do is actually to create a so-called main figure. Okay, first finish this and then go back to the question. So this main figure is a simple A4 page. So as you can see, the white part, which is a figure, it doesn't resize with the window, so it stays constant, and therefore it is basically a fixed dimension of this figure. And we can now add axes to this A4 page to collect the graphs that we created. For example, and now we first have to create and define a position where this figure that we want to export should be located. And to that reason, there is a grid that defines the axes or the position, the possible positions of this A4 page. Let's say in this case we have 4 rows and 3 columns, and you see directly what I mean by that once I created an axis. So we can just click on this plus sign. You can see we just create axis by axis. We have in total 3 columns, and then if we continue, we have 4 rows, which then determines the size of the axis. If we want to have bigger graphs, then we have 2 possibilities. Either we leave the grid as it is and we define the row span. So for example, we could say, okay, since we have 4 rows, we want to span this axis half of the page, and then maybe 2 column spans out of 3. And when we click yes, add, then it creates obviously a bigger axis. And this cannot be changed sadly afterwards. That is a bit annoying, but so we define first an axis and then we export a graph from the main window to this figure. And how to do that? I want to go with a smaller one. So we go like that. 1 by 1. We create three axes, and then if we go here to right click on any chart that we have, we can see that there is this 2 main figure, and F1 indicates figure 1. And then since we created already some axes, it has the name axis 8A, so labeling, and then if we put export, then it exports this this chart to the main figure. This is not interactive anymore, yeah? So this is kind of static. So whatever we do now here to this figure doesn't affect the figure that we have exported. We can mix it up with different kind of chart types and then collect these data, yeah? And the main advantage is really, because probably everyone who wrote a master thesis or PhD thesis was annoyed by aligning different subplots to each other in Adobe Illustrator, in the CorelDRAW, or whatever software you're using. And with that, you get a perfectly aligned set of figures. Yeah? It may be sometimes challenging to know the size that you want to use the chart for before you actually exported it. But I think once you're using it, you get used to it. You can, of course, change some basic things here as well, which can be done here by using this this little icon here. We can click on it and then select an axis, and then we can still change the labeling of the axis. We can also change the limits, and we can add the title. We can just apply to it, and these things we can change, of course, but we cannot change something like, okay, adapt all the charts as the same axis limits and so on. And you can, of course, also mix different grids. So let's say we want to add a smaller one. We can do like that, simply changing the number of columns, since then the A4 page is divided into multiple columns or more columns, and then allows us to create smaller axes. Yeah, and then like this you can build up. There's also not only the possibility to have one, you can have many main figures as you want. Yeah, and then you can of course also export them to a PDF and then work further on them. Because if you want to add a Western blot or whatever, another kind of chart, then you of course need to add them in a different software. Okay, I think this main figure has been proven to be relatively useful. I'm basically using it always to export charts, just to make sure that the size, the font size, and everything is aligned and equal between these the figures that I export. Okay, good. And then there was some question. Trying to find the chat. How can we plot the mean of the okay, this I think will be covered later. So you just want to calculate the mean of 3 different replicates, I guess. I think this we will cover later. Okay, I will first go on. Okay. If I forget to answer this, please remind me later. Okay, so I think Daniel has a question.
Yeah, my question was just now also asked in the chat. It seems like the lattice of your box plots of the axis were reaching into the letter into the F1, I think. Yeah, into figure F. And is there a way to change that, or are we stuck with that until we use a different software to realign everything?
Yeah, I would guess most of the times you're stuck with that. There is a chance to increase the size of the space. So like that. Let's maybe put 5. So you can change it, but you can only change the overall space between the axes. You know, you cannot change it for one particular axis. And sadly, there's also not a way to rotate for a certain axis at the moment. Yeah. So probably you would end up with doing this in Adobe Illustrator. So I mean, don't expect publication ready, but maybe close to publication ready. I guess.
Okay, thanks.
Yeah. It's the same question. Okay, perfect.
So that will be added in the future, if I understand right.
I don't think so, to be honest. So changing and rotating different ticks labels for charts, I think this is, since it's a bit more of in my free time when I'm doing this, I think it's
maybe a bit excessive. Yeah.
Yeah, but if you want to, you can contribute actually also to the code, of course, and do things. Okay. So then I want to just briefly mention that cover these other chart types and what is the difference. For example, I mean, we said already that if you want to plot the distribution of values, then we can use a box plot. If you want to have a more refined way of plotting this distribution, I think you may have seen this violin chart with estimated density curve to plot this line. Or we can use, which is actually recommended for very large data sets, so speaking 10,000, is the so-called Boxen plots, which is very similar to a box plot, but divides the data simply into more separate boxes. It's called a Boxen plot, therefore, and it's actually often able to visualize differences more at the tail of the distribution. So for very large data set, it's recommended to use this one. I want to make one point because this is often asked. You can always check the data of the box plot by right-clicking and say then box plot data, and then you can see the data that is used actually to plot the box plot. And you can either do to clipboard and then you can paste it into Excel, or you can add it as a new data frame. You demonstrate to clipboard, and then you simply go to Excel and then paste the data, and then you have basically the data that was calculated to plot the box plot. Okay, then I want to cover another type of plot, which is called the count plot. And with that, I want to introduce some of the filterings that we can apply in the software. And just to start with, if we have these category values and we put them into categories, then we get by default this so-called count plot. And this visualizes on the y-axis here the different categories. In this case, we have minus and plus. And for this particular data set, we have minus, we have 3,239 and 43 that are significant. And you may wonder why this is now plotted 2 times here, but you will see it if we combine it with multiple ones, then we get all the combinations and the counts. So in this case, it would say minus minus, that would be not significant in that comparison and not significant in this comparison. And then you can go on, this is significant in the red one, not in the blue, and so on. And then the right panel sums up all the sum of all these counts. No? And that allows you to relatively quickly see an overlap between significant hits or any other kind of filtering that you want to have. Usefully, you can always go to these categories and then go show subset, and then you can see the data that are basically associated now with these 30 proteins in this case. No? So this is actually able to combine multiple things. At some point, this gets very messy, but I want to show you just one example, because as I told you, we are very interested in mitochondrial proteins. What we want to use in this case is we want to take advantage of the so-called gene ontology annotations. And I hope many of you have heard of them already. These are annotations that are categorized or hierarchical annotations of genes that are divided into biological process, which is handling here BP, molecular function, and cellular component. And cellular component, for example, offers information about where a protein or a gene might be located. And we can use that actually to find, let's say, mitochondria proteins. And again, this is just as an example. You can use any kind of data. And sorry, I was a bit fast. I clicked on right-click the context menu, and then I opened the categorical filter. There's also the possibility to click on this filter to do the same, or you can take that and drag it onto the filter icon, and this will be all end up in the same window. What is a category now compared to a string? So for example, if you use Excel and you search for a string, then this is simple as string search. And if you had, imagine two categories which are called inner membrane and outer membrane, and you search for membrane, then you would find both. But the category search and filter is actually meant to find the distinct word. And as you can think of, I've actually prepared. Okay. I had prepared like a small slide for that. So imagine you have something like this, where you have a gene ontology, which usually have some ID, and you have them in your table split by a semicolon, and this is called a split string. And then you have multiple ones. And what a categorical filter would be, it would really find only this distinct category. While, as I said already, a text search might find multiple ones, yeah, because they have the same substring in them. And therefore, as you can see here in this filtering, you can see all these different or found categories based on this split string. So the split string is here the semicolon. If your proteomics facility or your data are of different type and you have them split by a different character, then you have to change this. But in this case, we have the semicolon for them. And every protein has multiple annotations, and we can now simply search for mitochondria. I guess it is like this. And then we click on it, and then we have two different options. So the first one would be to annotate the selection. Then the software would simply add a plus sign where it found this category in the column of cellular component. Or you can do a subset selection. Then it would create a new data frame with only containing the proteins that or genes that have this annotation. So in this case, we may only want to use this for annotating. We click yes, and then there is a new column created, which is called mitochondria in this GOCC. When InstantClue uses by default a plus sign to annotate this, and what we can then do is actually add this as well. We remove one here, and then in blue we have mitochondrion. So for example, if we want to find the number of significant proteins that have the annotation mitochondrion, we could look at ++ and then find this combination, and then we see these are the 15 proteins that are significant in the comparison that we are looking at, and they have the annotation mitochondria. And this filtering is is used relatively frequently, and therefore there are multiple options. For example, you can also have a combination of two. Yeah, so now I used molecular function in GOCC. Again, the underlying data doesn't matter. It doesn't have to be gene ontology. It can be any kind of string data, and then you can make combinations out of them and so on. Yeah, so the functionality is relatively extensive to filter these data. However, then we also have a different option, which is the so-called find strings. And this is really like in Excel, if you just search for something, since it will find a substring in the string, as you can see here. Now that the data that we looked at recently, this GOCC, were separated by a semicolon. You can see they are now listed simply like that. And we can now search, for example, for mitochondrion. Yeah, and then it shows us all the different rows where it found it. I just want to show you this when it may be advantage over the categorical filtering, because this can handle very easy multiple inputs. So for example, we could write mitochondrion, and then you hover over this, you can see that you can add multiple strings. Yeah. So for example, let's say we want to find I want to find proteins that are annotated by mitochondria and membrane in this case, then we can write it like this, and it will find both. And if we say now annotate matches by search string, then instead of using a plus sign, it will use mitochondrion or membrane to annotate matching data. Yeah. If we do that, let's quickly do. I think it's easier if you see the result. You can see we have the combination mitochondrion membrane and membrane. Yeah, here I have to. And there is no mitochondrial protein that doesn't have the annotation membrane, so therefore we don't see it. Yeah, but you see the point that just by using basically the strings, you can have actually multiple search results in one go. And usefully, if you are looking for a set of genes, what is also nice is that you can, let's say we have an Excel file, just making them up. We just use them as a copy. So if you go from Excel and you copy them and you paste them, then the software detects this and automatically splits them into these comma and then quotation marks. Yeah. So this allows you to relatively easy find a set of certain strings that you're looking for, and conveniently you can export them or import them from Excel. I don't see the chat anymore. I don't know if no, nothing. Okay. This filtering, of course, also works for numeric data, but very differently. If you have, as I said before here, the numeric float, and we use that to go here to filtering, then the filtering looks like this. We can define a subset of operators, how we want to filter these data. So for example, everything that is greater than 25 or something indicates by a blue font if this value matches somewhere. So if you put a value in, say 0.2, then basically, and it creates a black sign or it doesn't turn blue, then it indicates that it's somehow out of reach what you are filtering for. And what you can again do is to annotate matches and subsets. So meaning, again, you can filter and create a new subset. And a new subset, as you have seen, we have imported many data already today. You can always see here what is or the recent data frames. And lastly, there is another convenient way to filter data. Really the last one. 1 second, or then you maybe go on.
There was this other option. Wait, let me look how. N largest and N smallest. What does that do?
So basically it picks, if you now enter 10, then it would pick the 10 highest or smallest if you put large, smallest and largest. So you know the top 20%, top 20 or top 100 or so, you can subset with that.
Thanks.
Yeah. And lastly, as a filter option, you can use the spreadsheet light widget, and here you can simply right-click on a column header, and then we can define a range, let's say 25 to 27, and we confirm this by an enter, and you can see that now we have filtered the data. Based on this, you can remove this filtering by just clicking on it, and we can do this for multiple ones. In addition, we can also search for genes. Okay, it doesn't fit now, this filtering, but you can search for strings and for numeric data, nicely seeing already the result, how many you have. I think in the setting you can set, I'm not 100% sure if it should be considering this as and or, but at least you have a nice visual result already of your filtering. And then more importantly, you can either go here and say add as a new data frame, and then it adds this as a new data frame. Or on the other hand, if you close this window, it will recognize that you updated the data and ask you what you want to do. Let's say you want to update the data, and then you can choose if you want to basically override the data that you just have selected, or you want to create a new data set. So let's say no, and then you can see that we have here our filtering result now as a new data frame. Okay, I think this has been relatively extensive on the filtering. You will get used to it once you're using it, if you're using it. And yeah, okay. Then I think we don't have time for a break, so I will just continue. I think we have covered most of the things. One very important point that always comes is actually how to alter the font size and the ticks and so on. So maybe let's just plot one simple graph. So in the setting, I'm sorry, that was too fast. Here in the bottom right corner, you can choose from this drop-down menu. There is one which is called matplotlib, which is the library that is used to create these charts. And there you have the possibility to change the appearance of the graphs. For example, you can change that there is no box. Yeah, so let's say we want to turn off the box. It says axis binds right. This is the right side. We want to remove also the top one. And then let's say we want to have orange background in the figure. And let's say the white ticks which are going inside, which I learned is a very sensitive topic. A lot of people hate it. We can put it out by just selecting here. And then let's say we want to make it a bit bigger and so on. So you can change many things, the length, width of the ticks and so on. So, or if you want to display a grid, let's turn it on. Let me see a lot of change. So very, very nice graph now. Yeah, so you can basically alter a lot of things here to make it or to change it to what's your needs. I may change this at least. And then this is saved, yeah, so you don't have to worry about it. Once you reload InstantClue, then this will always be the same. Once you did that, turn off the, oops, I'm sorry, the grid. Okay, then we are more or less back to normal. Okay, then I want to introduce to you the quick select and the live graph. And to this end, I am using the hierarchical clustering. And the hierarchical clustering is a very powerful tool actually to visualize data. I see often in proteomics that people visualize basically the whole data set, which I think is not that useful. I have added here one example just to clarify the terminology. In this heat map, you always have two dendrograms, which are used to cluster the data, a column one and a row one. So the row one basically clusters the rows and the column one the columns. And you cannot change the order then. Yeah, if you do the clustering, then you have to live with the ordering. This is also a very common question. I will show you how to turn that off in a second. Then we have a representation of the data by color. So meaning we have some kind of color code. In this case, we have red for a high value and blue for a low value. And I just want to remind you, because I think heat maps and clusters are used extensively. Just as a reminder, it's very hard for the human brain to read quantitative value from color differences. Yeah, so a heat map is used usually to show a global trend, not a minor differences in the fold change. Yeah, because that is very hard for the human brain to interpret. So in our lab, we are using the heat map only on a subset of the data. Let's say we're having a genotype in a certain treatment, and then we do some kind of statistical test like an ANOVA, and we subset then the data, and and then we visualize this, because that gives you way more information than something like this, where you have just the clustering accordance to the abundance. Yeah, so just keep that in mind. So, and we often use the z-score, which we will also use in a second. And the z-score simply normalizes the data by taking every single data point that we have. It subtracts the mean and then divides it by the standard deviation. So you get basically a value that gives you how many standard deviations this value that you are looking at is away from the mean. Yeah? Be aware that this normalization is completely wiping out the information about the fold change. I've plotted you here 2 genes in log2 scale, which is here log2 difference of 6, a lot, a very high fold change, and a very minor fold change for gene 1. If you're using the z-score, because they're all going in the same direction and the standard deviation is very low, if you visualize them in the heat map, they are actually exactly the same. Yeah. So you cannot know the log2 fold change afterwards anymore. Yeah, if you don't save the data. So be aware that using the z-score, which is very commonly done in heat maps, completely ignores the fold change if you're looking at proteomics data. Yeah, just as an introduction. And what we want to do here is we want to use this tutorial data too to visualize that. And what we can do is, since these are the intensities, so they are not z-score transformed, we first get started with how this looks. So basically you just take the columns that you want to use and put them to the numeric receiver box. You click on the heat map and there you have it. Yeah. The labeling is now a bit bigger because I recently changed it. So if you see weird styling, then you may change that again. And what you can see nicely here is the row dendrogram and here the column dendrogram. You can see in this case that we really have a difference between replicate 1 and the other 2 replicates. Yeah? So this replicate 1 clusters away from the other replicates. Yeah? But here we are looking at all the data, and in addition, we are looking at an IP. Nevertheless, let's go through the functions of this heat map. So since this row dendrogram is calculated based on a distance measure, and this is by default the Euclidean distance, and you can read up how to calculate that. And this is then used to define how similar 2 proteins behave over the samples that we have measured. If you want to turn that off, you can go to the settings or change that and go here, and then we have different ways or different parameters that we can change. For example, we have the Euclidean distance, and the word of warning in InstantClue by default, it before it plots a hierarchical clustering, it will remove every row that contains an NaN. That is way faster to calculate, at least for this Euclidean distance. If you don't want that, if you want to show them, then you can enable this Euclidean distance, just that you know. But if we want to ignore the column clustering, we can just set the metric to none. And if we then replot the graph, then you can see that this is omitted and you get the ordering as we had put the data in here. Yeah. Okay, and on the other hand, you get these colorings here on the left side, which then is labeled with C1 for cluster and C2. And this is basically since the software tries to identify certain clusters. Yeah. And what we can do is actually we can click here and then move this line, and then it refines the clustering or it doesn't. Yeah. And that is actually but up to you what you want to use and what you are looking at. But I'm just telling you, you can basically define certain clusters based on your needs, and then you can just go right click and say export this one cluster ID, and then you have the annotations. Yeah. Okay. The functionality that I showed you for the for the scatter plot is also available here. So we can go for tooltip and then screen through the hierarchical clustering. If we take the G names and put them to labels, nothing happens. And that is because if we now annotated all the rows, it would be just a black bar because there are too many. So what it does, if we zoom in, yeah, then at some point, if a certain limit is reached, then it shows the different gene labels here on the side. We haven't spoken about these, but I think they're relatively intuitive. You can use also the panning, and then you can go through your hierarchical clustering. Most importantly, and I think this is one of the mostly used features, is that you can export this hierarchical clustering to Excel, so it's completely mirrored. So what you can do is you can do to Excel file. We just name it. We can save it. I don't see my file. And then once it's exported, you can see that now the export is basically mirroring exactly the colors as well as the clusters here of this heat map. Yeah? So that is quite nice. Once you have done it and you want to explore the data, then you can simply go through to the Excel file if you need more information. However, you can also get more information within InstantClue. Let me just replot that. And to this end, I want to introduce you this quick select and the live graph. And the quick select, for example, is a way to get as well any information. You can only put one column. So what I did is I just dragged and dropped here the G names, which then gives me the option to either throw the raw values, which means all the individual ones, or the unique values, which is similar to the categories we explored recently. And if I now go over this heat map, you can see on the left side that the G names change, yeah, and you can see what is the underlying. You can use this also to find your row of interest. Let's say we are very interested in this one, we can just type it, and then once we are there, it's actually indicated by this red scatter plot. Huh? And what I want to show you as well is that we can not only use G names, so meaning a single string, but as mentioned before, we can also utilize the categorical annotations. In this case, maybe the GOCC. We do the same. We want to have only the unique values, you can see, as in the filtering. And then let's say rewrite mitochondrion, for example. And then we go over this, and then it's not only one point anymore that is below one row in this quick select, but rather all the proteins that have this annotation. So you can really quickly see if there's a systematic accumulation of a certain pathway or geoter within the cluster that you are interested in. Was this to the heat map? This if we sorry, I'm reading. I don't know if you see it actually. Can we add the p-values as symbols in the plot? Was it
not in this whole big one, but I have seen publications that you have like the heat map, and then if this is real significant difference, then they add the stars to each of the genes, like to have a big overview of the genes that are significant.
So what you can do is, at least here, you can add a color column for either you plot numeric values for the p-values, minus log 10 transform, or you just indicate the significance. So for example, as we have here the plus signs for significance, what you could do in this plot at least is to add it to color. And then here it indicates by this blue line where the significant ones are. As I said before, I recommend usually to plot in a heat map a subset of all the data. Now we have a small one and it's okay, but for larger ones, it's good to do this filtering before, let's say, a p-value cutoff of an ANOVA or so, and then you plot all the ones that are significant. You can use this this color encoding for everything. Again, you can use also the mitochondrial annotations and so on. Yeah, but in this case, I would use this one. I hope you can use also numeric values. I just used intensity, but since p-value is also just numeric, you can also visualize basically the real p-value numbers by color using this. Again, I use simply drag and drop to the color and then it's added. However, you cannot mix categorical and numeric values in this case. Then, just to go on. So, I told you now a bit about this quick select. You can use this to search for genes, to search for GO terms, to do quite a lot of stuff. Just one thing that I think is useful, and I think in the online version this crashes, so maybe wait for the new one. What you can do is that you select multiple ones, and you can see how they are now added here to this hierarchical clustering. And what you can do is you can export this. So let's say you have a set of values, let's call them gene names, and that you always want to replot in every plot. You can save this as a list and then always upload it, and even after closing InstantClue. And this allows you to always have the same colors because each value is associated with the color. And then you can relatively quickly annotate your set of genes. Yeah, as you can see here now in this heat map, we can see, but if we change it to a scatter plot, these are actually saved and they are also annotated here. Yeah, and they will always be annotated. Yeah, in all the plots that we are doing. Yeah. Okay. Yeah. And then in addition to this, we can even extend the degree of data that we can extract from hierarchical clustering by, for example, we could use this one. And sorry, and I'm talking about the live graph. And this is a different panel, and this can visualize also data. So let's say we drag and drop all the intensity values, really doesn't matter what. So if I go now over the hierarchical clustering on the left hand, you can see, depending on data that I'm hovering over, the live graph basically updates and shows you the data that are there. And this is, of course, also working for a scatter plot. Yeah. So if I want to see, let's assume we have a volcano plot and you want to see how this protein behaves in other samples, you can just put all the samples in the live graph, and then you go over these data and you can directly see how they behave. This is one way to if you have a two-dimensional plot, to add more data that you can read out while going over these things. Okay, and okay, so then I know I'm speeding a bit, but we have only 20 minutes left. I want to quickly talk about the so-called groupings and what you can do with that. And as you can see here, we have a data table which has 3 replicates. In this case, they are control, and then this is the mutant, the wild type. Since it's an IP of a protease called PAL, and we have a catalytic dead mutant. This is basically a way how to trap substrates. But what you can see is we have 3 different groups of 3 different replicates. And conveniently, often also from proteomics facilities, you get already upfront calculated all these values, or the statistics, but you can also do that in InstantClue. So what we can do, for example, is, or what we have to tell the software first, which groups belong to each other. Yeah, because it doesn't know that control 1 and 2 and 3, even though it's obvious, belong to each other. So we have to annotate so-called groupings. And what you can do is, sorry, I was fast. I just clicked on the groups, on the right click on the numeric floats, and then I select annotate groups, and then this window appears. And this allows us to define now several groups. So we want to define control, then this S270A and wild type. Yeah. And what we have several options. What we can do is we can just take them and drag them to the column and then write here control. Yeah. And then we can go on and go on and go on. Yeah. But it's way easier because the name already gives us an idea what could be the group. And there's functionality to extract this information. So I selected the columns that I want to use for the grouping. And then this is called group by split string. So because if we find now between the different sample names a common name like control or wild type, the software can automatically group them together. And you have different options. For example, you can define the string. So if your facility or you divide the name not by an underscore, but rather by a space or something, you can add this here. And then you can define certain parameters to do this split and to define the groups. You always can see a preview here. So in this case, we're starting on the left. We take the index 0, so it's starting with 0 indexing. And the number of splits that it can perform is infinity, so it splits everything. There is a detailed introduction in the GitHub pages actually how this works. But basically, what it does now, it splits the string by this underscore, then you have control in 1, and it takes the first index, so 0 index, which is in control and this one. So you can see the result. We just click yes, and we have control, Y type, and so on. And we just call it genotype. Huh? Of course, there are very different namings, conventions, and so on. So there are different options how to extract this data, and often it's useful to think about your naming, how you name the sample. So if we do this, then you can see here on the data preview that by color it indicates now these groups. And conveniently, if we plot now something like an hierarchical clustering, it will ask if you want to use this actually to visualize this grouping. Usually we would say yes, and you can see now that it plots the color that we have defined for these groupings on top of the rows. Yeah? And this is also true for a correlation matrix, which we have not yet touched. Oh, sorry, I should turn off on the clustering again for this one. Sorry. So this is correlation matrix, and you can see that it now annotates these groupings. It can not only be used for this plotting, but we can also actually calculate something with these groupings. And for example, we can go here on the context menu to groupings, and then there are a variety of tests. We can do n-way ANOVA, so really as many as you want to, pairwise tests, like a t-test, for example, using these groups, which then calculates for each row the t-test. This is statistical analysis of microarrays, which is commonly used, is equipped with a permutation-based FDR. I think it's a bit too much detail. But what we can do for t-test is we just click on t-test, select the grouping. We can define the reference. So if this group is in the comparison, then always compare to this one. We click OK, and then we immediately get the result of the t-test. Yeah? And this is for an ANOVA, you get then the p-value for all these groupings. So if you have 3 groups, like a genotype, a treatment, and a time component, you can do the ANOVA analysis with this, and then do the subset on the p-value, and then do the hierarchical clustering, which is very common a workflow for us. Yeah, so there's a question if you can do the grouping of 2. So you can add as many as you want to. Let's say we, just for the sake of example, we add now a replicate group, which would be then identical to just have another one like diet or so. So in this case, we are not doing the first index, but the second one, which is called 1. And then you get 1, 2, 3, 4, 5. Let's call it for the question die. And then you get the annotation here. And if we now plot, then it detects that there are 2 groups, and we can basically select both, and then both are shown. And we did a smaller mistake here because in the groupings, you can define here the color map, and you probably should use another one for another grouping. To show them, you can always do here grouping and then switch between them. So you can sadly not show both within one go in this case. If you export InstantClue session, export the InstantClue session or save it, then these groupings actually will be also stored. You can also, if the naming is the same, export the grouping separately. And yes, two-way ANOVA is then available if you go here, groupings, multiple groupings, and then it's called n-way ANOVA, and then you define first what is your ANOVA type, and then you can select then these two groupings, and it will calculate the ANOVA. Again, you can do 10-way ANOVA whatsoever, yeah? So all is in there. It becomes very difficult to interpret, but then you, in theory, you can do that, yeah? Just for the last couple of minutes, we can touch this dimensional reduction technique. Let's maybe start with the PCA, and there also the grouping comes in very handy. So what we can do is we can select all the values that we want to use for the PCA. So PCA is a dimensional reduction technique. Since we have now very high dimensional data sets, meaning we have thousands of proteins, and in this case 9 samples, the PCA tries to visualize this data set within the 2-dimensional plot without losing a lot of information. And information for your PCA is variation. So it tries to, you can envision it like it tries to build up a new coordinate system through all through this high-dimensional data, and tries to retain as much information as possible. By default, InstantClue calculates 3 components. They are usually ordered by the variance that they explain. And how you can do that is simply go here to the right click. We have not really touched this context menu, but there is one which says dimensional reduction, and we have multiple ones here. We have an LDA, which is a supervised version. So meaning it knows upfront the groupings, the PCA, UMAP, TSNE, ISOMAP, and so on. So several things. What we want to do is, in this case, maybe you use a PCA projection, and then you get the components here, and in brackets you get the variance that is explained by these components. Then we can do scatter plot. And then conveniently, what I want to show you can see here the groupings actually that we had defined before, and you can then use them to annotate them. You can just put them to color, and you can see what we have seen already in the clustering, that there's one replicate, since diet is no replicate, we can encode the replicate by the size, and you can see that replicate 1 here is clustering away from it, because this simply says that the variance between the replicates is higher than the biological one in this case. Okay, then as the last thing, what is really now common to use is the so-called UMAP, and we are also using that a lot, and the workflow that we are utilizing is based on, again, selecting the significant ones and then calculating the UMAP. And the UMAP is a way also that tries to reduce the number of dimensions and visualizes everything in the 2D plot. And what we can do, or maybe you read up, since we are running out of time, on the theory behind that. In the GitHub pages, I have added a link there from, I think it's from Google, where you can play around with the UMAP and get really nice feeling about the parameters that you can set, because there are a lot of parameters that you can define, and they affect basically the outcome a lot. And therefore, I highly encourage you to go to this to the GitHub pages, and then there is this link where you can play around with UMAP and understand it a bit better. And I just show you how you can do that. Again, you can just go here, click on UMAP, and then instead compared to the PCA, where we get basically per sample values, and we use the projection. In the UMAP, we get a value for each single protein, and this takes some time. You can see that it's still calculating in this case, or which is shown here, and it may take some time to be completed, but yeah. Okay. In the meantime, we can wait until. Is there any question that you have at the moment? How to use it? Is there anything totally unclear? Yeah.
It's unrelated, but if you do all this analysis, can you say the pipeline, and then if you replace the data set, then all the graphs are automatically generated in the same way?
No, sadly not, but I think this is one what we are trying to do. Yeah, to save a workflow, and then you can basically apply
Exactly.
it.
Because sometimes, I mean, you just change like one treatment or like one genotype, and then you want the same analysis again. So it would be really good.
Yeah, sadly it's not possible at the moment.
Okay.
Yeah. Okay, so now we calculated, or the UMAP was calculated. It says then component UMAP 1 and 2, and you get the representation of all the data. So now each individual point is a protein, and I'm just showing you how we usually use this to analyze it. Again, I said we first do some statistical tests like an ANOVA, and then we are calculating the UMAP, which is a very convenient way to summarize a lot of data into one single plot. And you can assume that the more similar the proteins behave over the samples, they will be in one cluster. Again, I recommend you to go to this Google page, because you will see that the distance between two clusters is really not that informative, and it depends a lot on these settings that you can tweak around. But in general, if it's in one cluster, then you can use it. You can consider they are very similar. So you can basically use this live graph, if you can see here, to go through these different clusters. In this case, we use the log2 intensities. If you use the z-score, then it becomes a bit more informative. But what I want to show you is that there is a way how to cluster these data. And in conjunction with human, the same group actually developed this HDB scan. What you have to know about the clustering is that it works on densities, so how close 2 points are in this 2-dimensional or multi-dimensional space, and then they are clustered based on this density function. And you can just simply click on it, and the theory behind it I cannot explain you at the moment, but what I can tell you is that it's very good in combination with the UMAP, because it finds really distinct clusters. Again, the clustering output depends on the settings. I refer you to the settings here, where you can set all these. So, for example, dimensional reduction. Here it allows you to set all the different settings for the UMAP, and in the clustering settings, it allows you to set all the different parameters for the HDB scan. The nice thing about this HDB scan is that it's a clustering that allows to assign noise. So you always have some noise in your data, and this is indicated by this -1. So we can turn it off, and then you can see all these distinct clusters here across this UMAP. And then what you can do is you can use these clusters to find, in the case of proteomics transcriptomics, it would be to find a certain pathway enriched in this case. And at the moment, I'm showing you this for 3 groups, but this can be really extended to numerous groups, to up to 20 groups. And it becomes, in the beginning, it is a bit tricky to get the right settings, but over time, you will get a feeling actually what's good for your data. And then it's a very dense representation of the data. And what I mean by that is that you have a lot of information in a single graph. And yeah, that is what I wanted to show you. At the end, I want to basically, tell you that we have some proteomics toolkits like a 1D enrichment, pulse Silex. These are very specific for some use cases in the proteomics. You can also do enrichment analysis of pathways and so on. One thing that I have not touched at all here is different plot types, X and Y. You can find a bit information in the GitHub, but just play around with it, actually. It's my recommendation. What may be useful for some of you is the k-means clustering. Please, again, I refer to the Internet for Literature, for the theory behind that. But I just want to mention that we have here for k-means clustering a convenient one-click solution. So you just click on it. You can define the number of settings against here in this widget, but it allows you to cluster the proteins. Again, you would use that on a set of significance or something like this. But since this is more general data visualization, I wanted to show you more different plot types. And I think I've talked enough. Please write me, or if you have any question, we can also Zoom again if you have problems. And I would be really happy if at least some of you would use the software. And thank you very much for joining, even though the weather was really gorgeous. And thank you again for this opportunity. And have a nice evening, I would say. If there's any question, of course, we can discuss.